Okay, let me start. Okay, thank you for the introduction, Jeffrey. Today I'm going to present the first paper in statistical genetics of mine. It was published in Genetic Epidemiology. This research is a joint work with my student Liu and our former colleague Qinlu. So the title of this talk is Set-Based Genetic Association and the Interaction Tests for Survival Outcomes based on weighted V statistics. So let me first give you some background. In GWAS and sequencing studies, there are two common testing strategies. First one is single marker testing. Second one is set-based testing. The single marker testing tests one genetic marker at a time. The genetic marker could be a SNP, could be a gene expression value. And single marker testing usually suffers from low power because a single marker usually has a tiny effect on the phenotype. And because there are hundreds of thousands of genetic markers across the genome, so if you do single marker testing, you suffer from large burden of multiple testing correction. The set-based testing tests multiple genetic markers in a knowledge-based set. For example, you can test multiple SNPs within a gene, or you can test multiple genes within a biological pathway. So by aggregating the effects of multiple markers, set-based testing can increase the power for detecting genetic association and interaction. And set-based testing also remarkably reduces the number of hypothesis tests in GWAS and the sequencing studies. And usually set-based tests make use of kernel methods to account for the correlations among the markers. And that is different from the regular chi-square tests, like the likelihood ratio test, word test, score test. Those tests do not account for the correlations among the markers. And the survival outcome is also very useful in genetic studies. First of all, the survival outcome is more informative than the commonly used case control outcome. So it leads to a higher power to detect association and the interactions. And there are papers showing that advantage. And also the survival outcome is more appropriate for looking for genetic markers that are predictive of disease risk. In the literature, there are already set-based survival tests. There are 4 major set-based survival tests. The first one is called coxKM. It's based on a kernel machine Cox regression. It uses Monte Carlo methods to calculate p-values. The second one is the global test. The test statistic of global test is a special case of coxKM. It is the coxKM statistic with linear kernel. Global test has 2 versions. One version uses the approximates the asymptotic null distribution by a normal distribution. And the second version uses a permutation approach to compute the p-value. The third test is called coxSCAT. coxSCAT is also a special case of coxKM. It's the coxKM with linear kernel. But the difference is that the coxSCAT calculates the p-value analytically. And the fourth test is called AFTKM. It's based on a kernel machine accelerated failure time model. And it also uses the Monte Carlo method to calculate the p-values. All those 4 tests have remarkable drawbacks. coxKM, AFTKM, and the permutation-based GT are computationally demanding because they use simulation or permutation to do the test. And the asymptotic version of GT does not control type 1 error well when the number of genetic markers is small. Because their asymptotic distribution is valid only when the number of genetic markers converges to infinity. coxKM and AFDKM do not control type on error well when the sample size is small. And you will see this in the later simulations. coxKM cannot control type on error when the adjustment covariates and the genetic markers are correlated. So if you have confounders, then coxKM cannot deal with confounding. To introduce our test, let me define some notations. L is the left truncation time, because our test can deal with left truncation. If there's no left truncation, just simply set L to be 0. T is the survival time, C is the censoring time, and U is the minimum of T and C. This delta is the indicator of event, and the counting process is denoted by NT. YT denotes the at-risk process. The genetic markers of interest are denoted by this G, and then we adjust for a bunch of covariates denoted by Z. Small n denotes the sample size. First, we developed a weighted V test for genetic association without adjusting for covariates. So this test is based on this weighted V statistic. This is a population version because it involves some unknown parameters. So in this weighted V statistic, this MI is the martingale residual, and the MI times MJ is the kernel of this weighted V. And MI-MJ is a phenotype similarity measure. And this F tilde GIGJ is the weight function of the weighted V. And it is a centered genetic similarity measure defined by this formula. So in this formula, this F is a kernel function. And the idea of this weighted V is that bigger phenotype similarity is weighted by bigger genetic similarity, and the smaller phenotype similarity is weighted by smaller genetic similarity. So that will lead to a larger value for V star if the alternative hypothesis is true. The choice of this F function, the kernel function, does not affect the type on error, but it affects the power of the test. And its choice depends on the type of genetic markers and the effect form of genetic markers. So if the genetic markers, their effects are expected to be normal or to be linear, then we can use this cross product kernel, also called linear kernel, as the F function. If G is a set of SNPs and they are not expected to have a linear effect, then we can use the IBS kernel as the F function. If G is a set of gene expression values, then we can use either polynomial kernel or Gaussian kernel as that F function. And finally, there is a universal kernel called Laplacian kernel that can be used anytime. And in this Laplacian kernel, this WK is a weight. For example, we can use the inverse variance of the kth marker as WK. So if you do that, then you basically weigh more the rare variant and weigh less the common variant. So this kind of kernel is very useful in sequencing studies where you have a lot of rare variants. Then to do the test, we need to know the asymptotic, we need to know the null distribution of the weighted V statistic. We prove that under the null hypothesis that T is independent of G, n times the weighted V statistic converges to this mixture of chi-square 1 distributions, where this lambda is the mean of the squared martingale residual, and these eta t's are the eigenvalues of the function F tilde. Then in practice, we need to get a sample version of the weighted V, so it's computable. To do that, we estimate the hazard function of survival time by the Nelson-Allen estimator, and we approximate the expectation in F tilde by the sample mean. Then the resulting sample version of V star has this quadratic form. where this M hat is a vector of martingale residual, I is identity matrix, J is an n by n matrix of elements 1 over n, and F is a matrix of elements F, Gi, Gj. And we also need to estimate the unknown parameters in the null distribution in order to perform the test. Here we estimate the lambda by the sample mean of the squared martingale residual. And to estimate the eigenvalue eta t, we do an eigen decomposition of this matrix, I minus J transpose F, I minus J, and get its eigenvalues eta t tilde. Then we can use eta t tilde divided by n to estimate the eigenvalue eta t. Then you plug in the estimator into the null distribution, you get a computable null distribution. And based on this, we can use Davis method to compute the p-value. Then we developed a weighted v-test for genetic association adjusting for covariates. In this test, we assume that under the null hypothesis, survival time given adjustment covariates follows a Cox model. And then the weighted v-test is based on this weighted v statistic. Here, this MZI is the martingale residual under the above Cox model. And this F tilde Z is a covariate centered genetic similarity. It is defined by this expression. And then we showed that under the null hypothesis that T is independent of G given Z, and G is also independent of Z, the n times the previous weighted v statistic converges to this mixture of chi-square 1 distribution. And this zeta is the mean of the squared martingale residual, and this nu t is the eigenvalue of the function f tilde z. When G is not independent of Z, as long as G satisfies a linear model with Z and F is the cross product kernel, then we can get the same asymptotic null distribution as above. So in other words, our test can handle linear confounding. Then the sample version of this weighted V is obtained through estimating the unknown parameters. So here we estimate the gamma, the regression coefficients in the Cox model by maximum partial likelihood estimator. And we estimate the baseline hazard by the Breslow estimator. And we replace the expectations in F tilde Z by the corresponding sample average. Then the resulting sample version of the weighted V has this quadratic form. So here M hat Z is the vector of martingale residuals. And this I minus H, this is like a projection operator. It projects everything onto the column space of this matrix Z tilde, whose ith row is 1 Zi transpose. Then we estimate the unknown parameter zeta by the sample mean of the martingale residual square. And to estimate this nu t, we do an eigen decomposition of this matrix I minus H transpose F I minus H, get its eigenvalues nu t tilde, and then nu t tilde divided by n is an estimator of this nu t. Then we plug in the estimator, we get a computable null distribution for the weighted V. Then we developed a weighted V test considering genetic heterogeneity. Sometimes the effects of genetic markers vary across subjects or subpopulation. This phenomenon is called genetic heterogeneity. To account for it, you just need to do one change. You just need to change this kernel function FGI by a heterogeneity weighted kernel function. So in this heterogeneity weighted kernel function, this kappa ij is a measure of subpopulation similarity between subject I and subject J. Then this change leads to two heterogeneity weighted V tests. So this VH is the heterogeneity weighted V test without adjusting for covariates. This VZH is the one adjusting for covariates. And we can obtain their null distribution in the same way as before. Then the subpopulations or population structure sometimes can be completely determined by a vector of observable variables. For example, if the subpopulations are just different sexes, then you can just use dummy variables to code the different sexes. But sometimes the subpopulations are latent. For example, the subpopulations are based on different genetic profiles. In that situation, we can use a vector of SNPs to represent the genetic background. But the vector of SNPs cannot determine the genetic background completely. You can only use the vector of SNPs to infer the genetic background. So the subpopulation similarity, its choice depends on the subpopulation structure. In general, you can use this unified kernel to measure the subpopulation similarity. So in this unified kernel, XIDS is the standardized version of the D component in X. Then if your subpopulation are different genetic backgrounds, then you can use a set of SNPs to represent genetic backgrounds. And in that situation, you can use the IBS kernel to measure the subpopulation similarity. If the subpopulations are observable subpopulations, and X is a vector of dummy variables coding the subpopulation, then you can use this subpopulation identity kernel to measure the subpopulation similarity. And finally, we developed a weighted V-test for gene-gene and the gene-environmental interaction. So here, one gene is denoted by G. It consists of SNPs. Each component of G is a dose of a SNP. It's the dose of the minor annuals for that SNP. And the second variable is either a gene or environment variable, and it's denoted by H. Under the null hypothesis of no interaction, we assume that survival time given G and H follows a Cox model with G and H as additive covariates. Then we define this martingale residual based on this Cox model, and then define this M hat as the vector of martingale residual. G tilde is the matrix of GIs, and the H tilde is the matrix of HIs. Then we use kernel functions F and K to represent the similarity in G and H between subject I and subject J. Then the weighted V-test for testing genetic also a genetic interaction is based on this weighted V statistic. So here this M hat transpose times M hat serves as a phenotype similarity measurement. So in this phenotype similarity measurement, we already removed the main effects of G and H from the survival time. And the middle part, this I minus O transpose F times K I minus O, this serves as a interaction similarity measurement. And then we can use the same way as before to obtain the null distribution of this weighted V-test. Then let me show you some simulation results. So in the simulation, the genetic markers are all SNPs, unless otherwise specified. Each SNP is generated from this binomial distribution. And in the simulation, we adjusted for two covariates. One is Bernoulli, the other is uniform. And we generated survival times from different Cox models, depending on the simulation scenario. And we also had a simulation assessing AFT-KM. In that simulation, we generated survival time from the accelerated failure time model. The kernel function AF is the cross-product kernel, unless when assessing size and the power of HWV and the coxKM, where the kernel function is the IBS kernel. This table shows the runtimes of 5 tests, including our test WV and the 4 existing set-based tests. So as you can see here, coxKM and AFT-KM are very, very slow. And I even didn't get the runtime for AFT-KM when sample size is 500 and 1000. It's just too slow to finish. And the WV, GT, and coxKM, their runtimes are comparable. They have comparable computational speeds. When the sample size is 1000, WV is the fastest. This figure shows the QQ plots of p-values for 4 tests under the null hypothesis. The 4 tests are WV, coxKM, GT, and coxKM. As we know, under the null hypothesis, the QQ plot of p-value should look like a uniform 0-1 distribution. And here you can see only the QQ plots for WV and coxKM look like uniform 0-1. The QQ plots for GT and coxKM do not look like normal 0-1, do not look like a uniform 0-1. And according to the QQ plots for GT and coxKM, you can see that these 2 tests are conservative, meaning that their type 1 error is smaller than the nominal level. So under the alternative hypothesis, these 2 tests will suffer from low power. And this figure shows the QQ plot of p-value for AFT-KM. And you can see it doesn't look like uniform 0-1. And according to this QQ plot, we can see the AFT-KM is anti-conservative, meaning that its type 1 error is bigger than the nominal level. So you will get more false positives than expected if you use AFT-KM. And then we compare the QQ plots of p-values between WV and coxKM under linear confounding. So this time the genetic markers are continuous variables. They were generated from this linear model with Z1 and Z2 as 2 covariates. So here the genetic markers mimic the gene expression values. And under linear confounding, you can see the QQ plot for WV still look like uniform 0-1. But the coxKM, its QQ plot looks very bad. And this QQ plot implies that the coxKM is very, very conservative under linear confounding. Then we compare the size and the power between WV and the coxKM in the absence of genetic heterogeneity. So according to this table, you can see that both tests control the type 1 error well around the nominal level, and the 2 tests have comparable power. Then we compared the power of HWV and the coxKM under genetic heterogeneity. So this time we consider 4 observable subpopulations coded by these 3 dummy variables, and we let the genetic effects vary across the 4 subpopulations. Under this situation, you can see both tests still control the type 1 error well, but the HWV is more powerful than coxKM because HWV can account for genetic heterogeneity. Then we compared the size and power of WVI and the likelihood ratio test for testing gene-gene interaction. So here the genetic markers within a gene are correlated. And as you can see in this table, both tests control the type 1 error well, and WVI is more powerful than likelihood ratio test because WVI use kernel methods to account for the correlation among the genetic markers, and the likelihood ratio test cannot do that. And finally, we did a simulation to assess the empirical size of HWV under stringent p-value threshold. In GWAS and the sequencing studies, we have a lot of hypothesis tests to do. So we need to use confounding methods or FDR methods to control for multiple comparisons. And those approaches will lead to stringent p-value thresholds. So here you can see that under stringent p-value threshold, the empirical size of HWV is still close to the nominal level. So this indicates that our test is still applicable in GWAS and the sequencing studies. Now let me show you a real application. The real data come from these two studies, the religious order study and the Rush Memory and Aging Project. Both are longitudinal studies of dementia. Outcome of interest is age to Alzheimer's disease onset. In the analysis, we removed the subjects who had Alzheimer's disease at baseline because they do not have age to AD information. And doing so leads to a left truncation. After quality control, the final data set contains 1,433 subjects, and 461 of them developed AD during the follow-up. So in the analysis, we did a gene-based GWAS. We group SNPs into genes, and in total, there are 21,330 genes. In the association test, we adjusted for the top 20 principal components of the genetic data, and we also adjusted for sex, education, and cohort. And we did the genome scan using HWV and WV. In HWV, we considered genetic heterogeneity across individual genome profiles, heterogeneity across education levels, and heterogeneity across sexes. And we use Benjamin-Hochberg method to control the false discovery rate under 10%. This table shows the significant genes found by the tests. So as you can see, each of the 4 tests found that only APOE and APOC1 are the 2 significant genes. And according to the p-values, you can see that when considered heterogeneity across education levels, the p-values for APOE and APOC1 are smaller compared to the p-values for WV. So this indicates that there may be an interaction between education and these 2 genes. And there is a question, that is whether the association of APOC1 is due to its LD with APOE, because the typed SNP of APOC1 is not far away from APOE, so these 2 genes are correlated. Then to assess whether APOC1 has independent association with H to AD, we tested its association, adjusting for APOE and the covariates we adjusted before. And we used these 4 tests to do the independent association checking. And from the result, you can see that considering heterogeneity due to education, the APOC1 still has an association with H to AD, even adjusting for APOE. So the p-value is 0.005. Then in conclusion, in this paper, we developed 3 sets of set-based genetic association and interaction tests. The first set is called WV. WV is the genetic association test with survival outcomes without adjusting for genetic heterogeneity. And the second set is HWV. This is the genetic association test considering genetic heterogeneity. And the third set of tests is called WVI. This is to test for gene-gene and gene-environment interaction. Through simulations, we found that WV is faster than existing set-based survival tests, and it controls type 1 error much, much better than the existing tests. HWV, as far as we know, is the first set-based survival test that can account for genetic heterogeneity. Under heterogeneity, it is more powerful than the existing tests because the existing tests cannot account for genetic heterogeneity. And the WVI is more powerful than the regular tests for testing gene-gene and gene-environment interaction when the genetic markers are correlated. And finally, we did some simulation to assess the robustness of WV against misspecification of the Null model. We generated survival time from AFT model but still apply the Cox model-based WV to the data. And surprisingly, we found that the type 1 error for WV is still controlled very well. It's still very close to the nominal level. So WV shows some robustness against the misspecification of the Null model. But I didn't show the results here due to the time limit. And finally, I want to acknowledge the grounds that supported this research in part, and also the ground that supported the two studies where the data come from, as well as the ground that supported GWAS, which prepared, archived, and distributed the genetic data we used. And here are the references for this talk. And that's the end. Thank you. Our test can still have decent powers in that situation compared to the genetic association test without considering genetic heterogeneity. Our test is still more powerful than those tests, even if the subpopulations are just inferred by some variables. But I didn't put those simulation results in this talk.
Are there any other questions? I'm sure if you want to find out the details, you can go to this the Genetic Epidemiology journal to find the article. There's more reading to do there.
Yeah, it's already published online.
Yeah. All right. If there isn't any more, I think we can call it a day and give you 12 minutes back.
Okay.
Thank you, Chenxi. Thank you, everybody, for being here.
Thanks, Chenxi.
Thank you, Jim.